Alright, so now let's move on to question 21 over here. Question 21 over here says that the enzyme telomerase prevents the loss of telomeres after many mitotic cell cycle. So basically, what are the telomeres? Telomeres are basically found at the end of the each sister chromatid. These are the telomeres, and they basically contain the non-coding DNA sequence. They contain the non-coding DNA sequence. So basically what is their significance? That with every cell division they shorten up या फिर they lose their part after every cell division, right? So it basically the presence of the telomeres prevents the loss of the vital genes or the coding genes of the cell. Prevents the loss of the vital genes of the cell, yes. Alright, so now basically they are saying that which cell needs to transcribe telomerase enzyme. So telomerase, they are saying in the question that it is that enzyme which basically prevents the loss of the telomeres. And we know that telomere basically shortens with every cell division, and telomerase enzyme is preventing it. So they say that which cell is needed, which cells need to transcribe more telomerase enzyme, right? So the cells which have more cell division, right? The cells which have more cell division, which the cells which will divide more are required to transcribe more telomerase enzyme, right? So we know that the stem cells are the undifferentiated cells, right? So they divide continuously to differentiate into a specific type of a cell. So yes, stem cells need to transcribe more telomerase enzyme, right? Then the activated memory B lymphocytes. We know that the memory B lymphocytes, basically they are the long-lived cells of the immune system, right? And they can divide multiple times in order to produce the antibodies. So yes, activated memory B lymphocyte also needs to transcribe more telomerase enzyme. Then if we come to the helper T lymphocytes, which are secreting cytokines, so basically they do not require continuous cell divisions. So helper T lymphocytes do not need to transcribe the telomerase enzymes, right? No, they do not have the need to transcribe more telomerase enzyme than usual. Alright, so the correct answer for this MCQ would be option B, which is 1 and 2 only. Alright, so question number 22 over here says that a polypeptide molecule contains the amino acid sequence of the glycine, leucine, lysine and the valine. Now they are saying that the table shows DNA triplets of these amino acid, which tRNA anticodon are needed for the synthesis of this polypeptide. So basically this is the sequence of the DNA triplet. Then from the DNA it will be a process of transcription, and the mRNA will be made, which will be having the codons, and then complementary to those codons will be the anticodon, which will be located on the transverse RNA, which is the tRNA, right? So we have to make the whole sequence, right? So how we will start over here? We need to have a prior knowledge of the bases, like adenine binds with the thymine in case of the DNA replication, and in case of the transcription, in case of the formation of the mRNA from DNA, adenine binds with the uracil, right? And guanine is a base which binds with the cytosine, right? Now let's make the sequence. Over here in the DNA it is CCC, so it will be complementary to the base G. So we will write G over here, then GAA. G will be complementary to C, and in the place of A you will write U, right? A, the base pairing of A will be with the U. Now there is TTT, which will be AAA, right? And CAA will be transcribed as, C will be transcribed as G, and A will be transcribed as UU. Right, now this is the mRNA sequence, which is the codon sequence, and now comes the anticodon sequence. Right, now for the anticodon sequence, let's look over here that G will be complementary to C. So let's see where is C in the option. Here are the three C's on the option which will be complementary to G base. Right, then in the leucine there is CUU. C will be complementary to G, right, and UU will be complementary to A. Then over here it is A will be complementary to U. All the A's, 3 A's will be complementary to 3 U's, and similarly goes for GUU, that G will be complementary to C and U will be complementary to A. So hence we have got our correct answer over here, which is option B. I hope that this MCQ makes sense to all of you. Now let's move on to question number 23 over here. Question number 23 over here says that the diagram shows a section of a strand of the DNA. Alright, so over here from the question they are saying that it is a DNA. So we know in the DNA the sugar, the pentose sugar is deoxyribose sugar. Deoxyribose sugar, right? This is a nitrogenous base. Nitrogenous base, right? And this over here are the phosphodiester bonds, right? And similarly this is what they have asked us in the question, that which type of bond is labeled X, right? They are saying that which type of bond is basically labeled X. So it is the phosphodiester bond, right? So the correct answer for this question is option D. Alright, so now let's move on to question number 24. Question 24 over here says that which row correctly describes the cytosine. So basically cytosine over here is a nitrogenous base, right? We have the two types of nitrogenous base. One is the purine bases and one are the pyrimidine bases. Pyrimidine bases, right? The purine bases are having a 2-ring structure and the pyrimidine bases are having a 1-ring structure. In the purine bases the examples are adenine and the guanine, and in the 1-ring structure the examples are cytosine, thymine and the uracil, right? And if we talk about the complementary base pairing, so adenine pairs with the thymine or the uracil and it makes 2 hydrogen bonds over here, right? It makes 2 hydrogen bonds. And if guanine pairs with the cytosine, so it makes the 3 hydrogen bonds, right? It makes the 3 hydrogen bonds. Now let's look at the question. Ring structure of the cytosine. So basically they are asking us about the ring structure of the cytosine. So we know that the ring structure of the cytosine is a single ring structure. Why? Because the cytosine is basically a pyrimidine base, right? Cytosine is basically a pyrimidine base, so its structure would be single ring, right? And then in the next row, in the next column, they have given us the number of hydrogen bonds it forms with its complementary base, right? So it would be the number of hydrogen bonds it is forming is 3 hydrogen bonds, right? And it is a type of a pyrimidine base. So hence the answer over here is option C. This was a simple MCQ. Now let's move on to the question 25. Question 25 over here is asking that during the production of the protein molecules only one strand from the DNA double helix is used. Which name is given to the DNA strand that is used to produce a new protein? So basically each DNA strand acts as a template for the formation of the new strand, right? हम लोग that's why we call the DNA replication also a semi-conservative process as well, because one parent strand या फिर one old strand is used over there. But over here in this question basically they are asking us about the production of a protein molecule. So basically the DNA strand act as a template for the formation of the new strand, right? So over here the name which is given to the DNA strand which is used to produce the new protein would be the template strand. If we talk about the other options, the leading and the lagging strand are produced during the DNA replication process, right? Which are often known as the Okazaki fragments, right? Which is basically joined together by the DNA ligase enzyme, if I am not wrong. Yes, the DNA ligase enzyme, right? So I hope that this MCQ is clear. Now let's move on to the question 26. Question 26 over here says that the diagram shows a longitudinal section of a phloem sieve tube with a companion cell. So this is basically the phloem sieve tube element. Phloem sieve tube element. And this is basically the companion cell. Companion cell basically supports the structure of the phloem sieve tube element, right? Where are the mitochondria located for the release of energy for the cotransport, right? So basically यहाँ पे we have to understand first of all that what is a cotransport. Cotransport is basically a type of the active transport, right? Where the energy which is used to pass the या फिर to transport the molecules comes from the gradient of the H positive ion, right? So basically the answer over here would be option B would be our correct answer, that companion cell have a comparatively higher ratio of mitochondria than the sieve tube element, right? Higher than the sieve tube element, right? So I hope that this is clear to all of you, this MCQ, that why we have selected option B, because the mitochondria content in the companion cell is much greater than the phloem sieve tube element. If you guys remember from your lecture, almost most of the phloem sieve tube is empty in order to facilitate the process of the translocation, the transport of the sucrose from the source and sink, source to the sink, right? So that's why most of the phloem sieve tube is empty. So most of the mitochondria are present in the companion cell, and majorly if we become more specific, so the mitochondria for the which provides the energy for the co-transport are specifically present in the companion cell. I hope that this is clear. Now let's move on to question 27. It is also from the transport in plants chapter. So now let's discuss this MCQ. Over here they are saying that the diagram shows a phloem sieve tube element and a companion cell that are involved in the translocation of the sucrose. Alright, which process correctly describes the translocation of sucrose through these cells? So basically what we have studied is that the process of the translocation of the sucrose mainly depends on the hydrogen ion concentration, right? Basically first of all hydrogen ion concentration is being built up, and then it provides the energy for the transport of the sucrose into the companion cell, right? It provides the energy for the transport of sucrose into companion cell, right? So basically the answer over here would be option B. Protons move from Y to Z by the active transport, and then this proton gradient provides the energy for the transport of the sucrose into the companion cells, right? I hope that this MCQ is clear. Now let's move on to question 28 over here. Question 28 over here is saying that which type of molecules are co-transported into the companion cells. So basically we all know that companion cells take up the sucrose, right? We all know that the companion cells take up the sucrose from the source and it delivers it to the sink, right? So basically sucrose is a disaccharide, right? It is a disaccharide, and from which monosaccharides the sucrose is made up of. Let me check. Yes, So basically the sucrose is made up of fructose and glucose, right? So basically what can be the correct answer of this question, that which type of molecules are co-transported into the companion cell? It may be the disaccharides, and the monomers, right? So option A would be our correct answer over here. Option A would be our correct answer over here. I hope that this is clear to all of you. Obviously, polysaccharides cannot be co-transported into the companion cells. Neither the polymers can be transported. We have not studied anything like this in the transport of plants, and neither of any of the other option makes sense, right? So now let's continue with question number 29 over here. Let's now have a look at question 29 over here. Question 29 says that the diagram shows a section through the human heart. Now this is a human heart, so let's label it first without looking at the question, that what basically the question is asking. This is A is the aorta. The heart labeling is very important to learn. You guys are now in the A levels and must know how to label the heart. It is very high yield for your examinations, right? B is the left atrium. Alright, this is the left atrium. Then over here, this is the left ventricle. And then the C is the right atrium, right? It is the right atrium. And over here the D is basically the pulmonary artery. I think this is the vena cava over here. Yes, these are the vena cava, superior and the inferior vena cava. Now they are basically saying in the question that which label is correct. So over here, A is the pulmonary artery. Is A the pulmonary artery? No. A is the aorta, right? B is the left ventricle? No. B is the left atrium. C is the right atrium? Yes, C is the right atrium. So definitely option C will be our correct answer. So it was very simple. Now let's move on to question 30. Question 30 over here is saying that the graph shows how the volume of the left ventricle changes during the one cardiac cycle. Alright, so basically this is the graph of the ventricular volume, right? Now let's see that what they are asking in the question, which point on the graph represents the start of the atrial systole. So let's discuss this graph one by one. Let's discuss the points on these graphs basically one by one. So basically the point A over here may be the ventricular systole, because you can see that the ventricular volume is basically decreasing, and it is almost coming to 40, right? And then after, at the point B, what is happening over here, that the volume is again increasing, which means that it is a diastole. The heart is going through a diastolic phase, right, where the ventricles are again being filled with the blood, right? And then this is the atrial systole part, where the where the atrias are contracting and they are pushing their blood into the ventricles, right? So the correct answer for this question is basically option C, right? The correct answer for this question is option C. Over here at this point in the graph, the atrial systole is being started, right? So I hope that this graph makes sense. Alright, so now let's move on to question 31 over here. It says that which statement is correct. Alright, so let's look at the statements one by one and let's try to figure out the correct answer. In the red blood cells, carbon dioxide can combine with the hemoglobin to form the hemoglobinic acid. No, this is not the correct statement. What happens is that the carbon dioxide reacts with the water to form the carbonic acid, right, which is basically H2CO3. This is the carbonic acid, which basically breaks into H positive ion and it breaks into HCO3 negative ion, right? So it is in option A is incorrect. So let's now see option B. It says that carbonic anhydrase is an enzyme that catalyzes the reaction between carbon dioxide and water. Yes, this statement is absolutely correct, that carbonic anhydrase is basically an enzyme which is catalyzing this reaction between the carbon dioxide and water, which is happening in the red blood cell. So hence the correct answer for question 31 is option B. Now let's begin. Let's solve question 32. It says that which row is correct for an artery. So basically in an artery there are three layers. Inner layer, which is known as the tunica intima, the middle layer, which is known as tunica media, and the outer layer, which is known as the tunica externa. So from the notes and from the syllabus outlines, we know that the inner layer is made up of the smooth layer of the squamous cells. The inner layer of the artery is basically made up of the squamous cells. The endodermis cells are basically found in the endodermal layers of the plant cells, not the animal cells. Alright, endodermis is not found in the animal cells. It is a specific feature of the plant cells, right? So smooth layer of squamous cells is found in the middle layer. Collagen, elastic fibers and smooth muscles are found, and in the outer layer collagen and elastic fibers are found. So the correct answer for question 32 is option C. Let's now move on to question 33 over here. Question 33 over here says that which row shows the features of the gas exchange surface that increases the diffusion of carbon dioxide and oxygen. So basically the rate of diffusion is very much high when there is a large surface area, right? Because the diffusion, basically the rate of diffusion, it is the rate of diffusion. So basically the rate of diffusion depends upon the surface area to volume ratio. We can say the surface area and the diffusion distance, right? If there is a large surface area, so the rate of diffusion will be more, right? More rate of diffusion will be there, and if there will be a short diffusion distance, then the diffusion distance will be short, so the rate of diffusion will be increased, right? There will be more efficient rate of diffusion. So now let's look at the options. Concentration gradient would be steep if you want to increase the rate of diffusion, right? So diffusion distance would be short so that more efficient gas exchange may take place. So hence the answer to question 33 is option B. Let's now have a look at question 34, which is a very lengthy question as well and a lot tricky as well. We have to basically interpret the whole of the information which is given in the question and then conclude our correct answer, right? So let's start by reading this question. Basically in this question they say that the electron micrograph shows some of the airways in the gas exchange system of an insect and the respiring body cells that surround them. Alright, so basically over here they are saying that these structures which they are pointing over here, they are saying that these structures over here are basically the trachea, and each of these trachea is filled with the air which enters through tiny holes called spiracles. Each trachea is held open by spirals of the material called chitin. Alright, and then they are saying over here that these branches of the trachea are basically the tracheoles, which are the narrow tubes which branches from a wide trachea. They lie next to the body cells, and gaseous exchange occurs across them directly into the respiring body cells. Alright, so now let's see that what they are basically asking us in the question. They are saying that which statement describes the correct differences between the insect gas exchange system shown in the electron micrograph and the human gas exchange system. Basically they have given us the knowledge of the insect gas exchange system, and human gas exchange system is basically in our syllabus, right? Insect gas exchange system is not in our syllabus, but the information which is required to solve this particular question is basically given in the question. So let's read the statements one by one and let's see that which statement correctly describes the difference between the both of the gas exchange system of the insect and the human, right? Gas exchange occurs through the walls of the airways directly into the respiring body cells in the insect, but this does not occur in the humans. Yes, this is absolutely correct that In the humans, the gas exchange cannot occur through the walls of the airway. It occurs in through the alveolus, right? It occurs through the alveolus. And then this statement is correct. Now let's look at statement 2. There are spirals of chitin in the wall of the trachea and the insect to hold it open, but not in the humans. Yes, no. This statement is also correct, that chitin is not found in humans. Not found in humans. In place of the chitin, basically to keep the trachea open in the humans, we have the C-shaped cartilage, right? I hope you remember its function. C-shaped cartilage, right? It prevents the collapse of the trachea under high pressure. They say that there is more than one trachea in the gas exchange system of the insect, but only one in human. Yes, over here in this question they are saying that tracheoles, which are the narrow tube which branch from the wide trachea. Basically they are saying that there is the trachea and there are the branches of the trachea as well, which are known as the tracheoles in the insect's gas exchange system, right? But there is only one trachea in the human. Yes, this statement is also correct. So hence the answer to this question is option A. All of these statements 1, 2 and 3 are correct. So I hope that this MCQ is clear to all of you. It was a bit tricky MCQ, so you should have an active brain while solving these type of MCQs. Let's now move on to question number 35 over here. Question 35 over here states that which row is correct for the wall of the trachea and the wall of the bronchus. First of all, there comes the trachea, then there comes the bronchus, and that bronchus is basically divided into a lot of bronchioles, right? So basically they are saying over here that what is common in both, basically what is common in the trachea and what is common in the wall of the bronchus. They are asking us about the common structures, right? So the cartilage is found in the trachea, yes, and as well as the bronchus as well. In trachea the C-shaped cartilage is found, right? And in the bronchus there are the irregular pieces of the cartilage which are found. Smooth muscles, yes, in both the trachea and the bronchus the smooth muscles are found. Goblet cells are, yes, also found in the epithelium of the trachea as well as the bronchus. So hence the answer over here to this question is option A. Alright, in many questions in the past exam it has also come about the bronchioles as well. So the key points which you have to remember for the bronchioles is that the bronchioles have no cartilage. It is a very high yield, they are the very high yield points which I am telling you over here, that bronchioles have no cartilage and they have a ciliated epithelium without the goblet cells. Alright, so it is very important for you to remember for your exams that bronchioles have no cartilage as well as they have the ciliated epithelium without the goblet cells. Alright, so this is a very high yield, these are the very high yield points to remember about the bronchioles as well. Now let's move on to question number 36 over here. Question 36 over here basically says that a bacterial pathogen produces a protein that acts as a toxin. Alright, this toxin is harmful to humans. Alright, we can understand this statement. Scientists are developing monoclonal antibodies that can be used to detect the presence of the toxin in the body so that the early treatment can be given. Which statement describes the steps in the development of these monoclonal antibodies? So basically over here you must have a prior knowledge about the steps which are involved in the development of the antibodies. Let me open a PDF over here where I have basically written the steps of the monoclonal antibody production. Alright, so I have opened my notes over here which are basically of the immunity. I made it, I guess, a few months ago when I was recording the topical MCQs of the immunity. You guys can check the topical MCQs of the immunity as well on my channel. Alright, so now let's discuss the steps to synthesize the monoclonal antibodies, and then after we understand these steps, it will become very easy for us to solve this MCQ, right? So basically what happens is that first of all the desired antigen against which the monoclonal antibody has to be synthesized is injected into the mouth, right? The desired antigen is being injected into the body of the mouse, and what will happen that due to that desired antigen the immune response will occur against that antigen, right? And the B lymphocytes will differentiate into plasma cells, and that plasma cells will produce the antibodies, right? So what we will do that these plasma cells which are produced in response to that antigen which is dangerous to the human body against which the monoclonal antibody is to be produced will be extracted, right? The plasma cell will be extracted from the spleen of the mouse, all right? So now these cell solution will have the plasma cells that will secrete the desired antibodies, right? And which do not also, all right? So what we will do that the plasma cells will combine with the myeloma cells which are the cancerous white blood cells, and they will produce the hybridoma cells, right? The plasma cells combining with the myeloma cells which are the cancerous cells will be called the hybridoma cells. Now what will happen that these hybridoma cells secretes the desired antibodies are identified using screening. Right now we will do a process of the screening where the hybridoma cells which will be secreting the desired antibodies will be identified using screening. Why? Because when we have extracted the plasma cells from the spleen of the mouse, these cell solution contains the plasma cells that secretes the desired antibodies and which do not. So that's why the screening is important. Then what will happen? The hybridoma cell is creating the desired antibodies and are separated and cultured to produce the large number of monoclonal antibodies, right? Now the desired hybridoma cells will be separated and produce the monoclonal antibodies, right? So now let's see that what they are basically saying in the question. They are saying to describe the steps which are involved in the development of the monoclonal antibodies. Let's read statement 1 over here. It says that the toxin protein is injected into a mouse and triggers mitosis of the specific B lymphocyte. Yes, this statement is correct because this toxin protein is basically acting as an antigen. It will go into the body of the mouse and will trigger the differentiation of the B lymphocytes, as it is discussed over here that the antigen will cause an immune response and it will activate a specific B lymphocyte which will differentiate into the plasma cell. So yes, this statement is absolutely correct. Right now, statement 2 says antibodies are collected from the spleen of the mouse and fuse with the myeloma cells. No, this statement is incorrect. Why? Because antibodies are not collected, the plasma cells are collected, right? Similarly, statement 3 is saying a hybridoma cells produces many antibodies with a variety of different variable regions. No, this statement is incorrect because only hybridoma cells only produce this specific type of monoclonal antibodies, and those specific type of the monoclonal antibodies will be used to treat the patient, right? So basically the hybridoma cells which will produce the desired antibodies will be used at a large scale to produce more monoclonal antibodies, right? So only the statement 1 is correct over here. So hence the answer to question 36 is option B, 1 only. Similarly, let's now move on to question number 37, which says that which statement are correct about the penicillin. Now if I talk about the penicillin, so basically it is a very popular antibiotic. It is a very popular antibiotic which is basically made from the fungus. Now they are saying that which statement is correct. We have to choose the correct statements for the penicillin. It is harmful to the prokaryotic cells. Yes, it is definitely harmful for the prokaryotic cells, which are the bacterial cells. What happens is that the penicillin basically interrupts the cell wall synthesis of the prokaryotic cells or the bacterial cells. It interrupts the cell wall synthesis of the bacterial cells and it disintegrates the cell wall basically. So yes, it disrupts the cell wall synthesis as well. It is harmful to the prokaryotic cells also. it becomes less effective with the regular use. Definitely we have studied about the antibiotic resistance, and we know that the bacteria develop the antibiotic resistance if the antibiotic is used in a very high dose and used more frequently, so the antibiotic resistance can be developed. So yes, this statement is also correct. So all the three statements are correct for the penicillin. So hence the answer to this question would be option A. 1, 2 and 3, all statements are correct. Alright, so now let's move on to the question 38 over here. Question 38 basically says that why is passive immunity effective for only short term? So basically there are two types of immunity. One is the active immunity and the other one is the passive immunity. In the active immunity, basically the weakened antigen is injected into the body and the B lymphocytes differentiate and produce the plasma cells, which in turn produces the antibodies. Also the active immunity is long lived because also the memory B lymphocytes are also produced, right? And in the passive immunity, basically the antibodies itself are being injected into the body, and these antibodies we have studied in the chapter that they are proteinaceous in nature and they are very short lived and they die after some time. They die or they become digested by the body cells, right? So we can say over here that the answer should be antibodies are rapidly broken down. Option A is our correct answer. That's why the passive immunity is effective for only a short time, because the antibodies which are given are broken down and also no memory cells are produced to prevent against the infection if the same antigen enters in the future as well. So now let's move on to question 39 over here. It says that which row is correct for the control or prevention method of each disease. TB, which is known as the tuberculosis, which is basically spread due to the coughing, right? Because it is spread due to the coughing, the water droplets in the cuff. So basically it can be traced via the it can be prevented or controlled via the contact tracing. The person who is having the TB, that person has been contacted with uh, how many people since he is coughing should be traced back, so that those people can also be tested for the TB and they may be given the precautionary measures or the treatment so that they may not spread the TB to the unaffected person, right? So I hope that this is making sense to you. Then the malaria. We know that malaria has a very famous vector, which is the female anopheles mosquito. So we can prevent the malaria by the destruction of the female anopheles mosquito, which is the vector of it. Now the cholera. Cholera basically is spread due to the improper sanitation of the water, right? Improper sanitation or the poor sewage treatment, right? Poor sewage treatment. So these are the major cause of the spread of the cholera disease. So basically we can prevent it or control it by the chlorination of the water. So hence the correct answer over here would be option C. Alright, so now let's move on to the last question of this paper, which is question number 40. It says that peptidoglycan is stained purple by the chemical crystal violet. Basically, what is the peptidoglycan? If you guys recall, so peptidoglycan is basically a compound which makes up the cell wall of the bacteria, right? Which makes up the cell wall of the bacteria. Now they are saying that which cell would stain purple in the presence of the crystal violet. Obviously, a bacterial cell would stain purple in the presence of the crystal violet stain, because the peptidoglycan is basically being stained purple, right? And peptidoglycan is found in the cell wall of the bacteria cell. Alright, so over here if we look at the options, so we can clearly judge that Vibrio cholera is the bacteria over here, right? Vibrio cholera bacteria causes the infectious disease which is known as the cholera, right? Which is basically spread due to the improper sanitation or the poor sewage treatment. So Vibrio cholerae is a bacteria which causes the infectious disease cholera, and because it is a bacteria, so it will be having the peptidoglycan in its cell wall. So hence the correct answer for question 40 is option B, which is the vibrio cholera cells. Finally, this was the last question, and this wraps up our paper of winter 2024, paper 1 2. I hope that you guys enjoyed solving this past paper with me, and if you guys did, then do consider subscribing this channel. Your support and your prayers means a lot to me. Thank you so much, everyone. Do prepare well for your upcoming examinations, and best of luck from my side.